All right, let's talk about the single most important chemical that gets your brain firing on all cylinders: glutamate. You can think of it as the master switch, the thing that powers pretty much every single thought, action, and memory you have. So let's dive in and see how this whole system works.
I mean, have you ever really thought about it? What is physically happening inside your head when you learn something new? What's the actual process that makes a memory or a new skill or even just a random fact stick?
Well, the answer to that question really begins with glutamate. If your brain were a high-performance engine, glutamate would be the accelerator pedal. It is by far the most abundant excitatory neurotransmitter we have, and its job is to get neurons to fire up and talk to each other. It's the go signal for your entire nervous system.
Okay, but to really get a handle on how this accelerator works, we first have to understand its life cycle. Where does it come from and where does it go? Because it's actually part of this fascinating, super efficient recycling program.
This image right here perfectly captures this incredible partnership between our neurons, the star communicators, and their essential support crew, the glial cells. It's a constant dance. See, glutamate gets made in the neuron, it's released into the synapse to pass on a message, and then, and this is crucial, it's immediately cleaned up by those glial cells to be recycled.
So let's break that down into 4 simple steps. First, the neuron makes glutamate and packs it up, ready for launch. Step 2, when the neuron fires, boom, it's released. Third, and this is super important, it can't just hang around in the synapse, so the glial cells jump in and suck it up using these special transporters. And finally, those glial cells convert it into an inactive form called glutamine, ship it back to the neuron, and the whole process starts over. It's a perfect, sustainable loop.
Okay, so glutamate is the message, but how does the next neuron actually hear it? The signal gets picked up by these specialized proteins called receptors, and it turns out there are a couple of different kinds of docking stations for this one very important message. So the first family of receptors we're going to look at are the fast ones, the ionotropic receptors. Now this table shows 3 main types, but for our story, we're going to zero in on the 2 heavy hitters. AMPA, which is kind of like the brain's simple, reliable workhorse, and NMDA, which is a much, much smarter and more complicated kind of switch. AMPA receptors are the most common ones, by far. Just think of them as a basic light switch. When glutamate docks with it, a channel instantly opens, sodium ions flood in, and the neuron gets excited. It's fast, it's simple, and it's responsible for the vast majority of speedy communication in your brain. On, off. Simple as that.
But the NMDA receptor, this is where things get really, really cool. This isn't just a simple light switch. No, it's more like a smart switch, one that needs 2 very specific conditions to be met before it even considers turning on. And this illustration shows that dual control system beautifully. For an NMDA receptor to activate, it needs glutamate to bind to it, sure, but that's not enough. It also needs the neuron to already be a little bit buzzed, already partially activated by other signals. We call that depolarization. This is why scientists call it a coincidence detector. It only fires when it senses that multiple things are happening all at once. So how in the world does it detect that coincidence? Well, the secret is a tiny little plug. You can see it in this graph. When the neuron is just resting, the NMDA channel is literally blocked by a magnesium ion. It's like a cork in a bottle. It doesn't matter how much glutamate shows up, nothing's getting through. But when the neuron starts to get excited from other inputs, that electrical charge actually shoves the magnesium plug out of the way, and voila, the channel is finally open. It's an absolutely brilliant voltage-sensitive lock.
And the complexity doesn't even stop there. I mean, look at this thing. The NMDA receptor is this incredibly sophisticated machine with all these different docking sites. It doesn't just need glutamate, it also needs a little helper molecule, a coagonist like glycine, to fully activate. It also has all these other spots that can be blocked by certain drugs, which is exactly why it's such a massive target for research into everything from anesthesia to mental health.
Okay, let's move on to the other major class of glutamate receptors. Now, these guys don't work like on-off switches at all. They're much more subtle. Think of them more like dimmer switches, the kind that can fine-tune a neuron's activity over a longer period of time.
So here's the key difference, side by side. Ionotropic receptors, like our AMPA and NMDA friends, are direct and fast. They literally are the ion channel. But these metatropic receptors, or mGluARs for short, they're indirect. They kick off this chain reaction of proteins inside the cell, which leads to slower, more widespread changes. It's really the difference between flipping a light switch and slowly, carefully turning up a dimmer.
And here's how it all comes together at a single connection point. This diagram is like our "you are here" map. You can see our light switches, AMPA and NMDA, on the receiving end of the neuron,
But check out where the dimmer switches, the mGluARs, are. They can be there too, but they're also often found on the sending neuron, where they act kind of like a volume knob, telling that neuron to release a little more or a little less glutamate next time. It's a beautiful feedback system that fine-tunes the whole conversation.
Okay, so we've got our accelerator pedal, we've got our light switches, and we've got our dimmer switches. Now let's finally connect all this incredible machinery back to our original question. How does any of this actually form a memory?
The answer lies in this powerful concept called synaptic plasticity. And it's a really simple but profound idea. The connections between your neurons are not fixed. They're not hardwired. They can get stronger or they can get weaker, all based on how much they're used. And this is the fundamental basis of all learning and memory.
So how does a connection get stronger? Well, imagine you're practicing a new skill. You're getting strong, repetitive firing in a specific pathway. This causes a lot of glutamate to be released, which unplugs all those smart NMDA receptors, letting a huge rush of calcium flood into the cell. That calcium surge is the trigger for what we call long-term potentiation, or LTP. It's basically a signal to the cell that says, hey, this connection is important. Let's install more AMPA light switches here to make it even stronger and more sensitive in the future. But here's the truly elegant part. The very same system can do the exact opposite. If you have weaker, less coordinated firing, you only get a little trickle of calcium through those same NMDA receptors. And this smaller, weaker signal is interpreted completely differently. It activates a whole other set of enzymes that do the reverse. They actually go in and remove AMPA receptors from the synapse. This is called long-term depression, or LTD, and it's how the brain weakens and prunes away the connections that aren't being used as much.
So, what's the big takeaway here? It's that the NMDA receptor, that beautiful little coincidence detector, is the master switch for learning. LTP strengthens the important connections, locking in our memories. And LDT clears out the noise by weakening the less important ones. It's this constant push and pull, this strengthening and weakening, that allows our brains to be so incredibly adaptive and to keep learning throughout our entire lives.
Which, of course, leaves us with one last, pretty big thought to chew on. If our memories, our skills, and maybe even our personalities are all just encoded in the ever-changing strength of these tiny connections, what does that really say about the nature of who we are? Something to think about. Thanks for joining me.